Thanks. Can everyone see it?
Mm-hmm.
Yeah.
It's
Oh,
just you're not using the PowerPoint.
Yeah.
thanks. Can everyone see it?
Mm-hmm.
Yeah.
It's
Oh,
just you're not using the PowerPoint. I mean, you're not using the presentation mode.
uh, how about now?
Yeah.
Yeah, I'll just briefly go through the things that I'm going to include in my introduction in the thesis and what data I have at the moment, and at the end I want to raise some of the issue that I'm facing at the moment. So I hope I think I can get some input before I move to my statistic analysis. And yeah, this is my final topic. It's about my master thesis, the investigation of small heat-shock protein expression under ER stress treated C2C12 myotubes. And a briefly introduction about the literature. Skeletal muscle basically have a lot of function, and it helps body to perform posture and locomotion and energy metabolism. It store a lot of glucose, and it basically maintain a lot of metabolic pathways. And in aging, basically there has a lot of factors like exercise, nutrition, and hormone regulation that leads to a decrease in general skeletal muscle size. And literature has linked that it will actually has an association with obesity, diabetes, or cancer, this kind of chronic diseases. And to maintain a lot of cell in the body, actually there has a system called proteostasis, which is help protein from unfold protein into a final folded proteins. And in this system, basically you have three main branches, will be protein synthesis and protein and folding, protein conformation and maintenance, and protein degradation. And this system will basically help most of the cells in the body to throw their 3D structure before they send to their final destination and work and do their function. And in aging, actually, it has been reported that loss of proteostasis is a hallmark of aging, and it, and literature has recently showed that actually loss of proteostasis is linked to some of the muscle-related diseases like sarcopenia and myopathy. And within the proteostasis system, a very important protein called chaperone is basically help to regulate the proteostasis networking system. In general, chaperone has involved in all types of proteostasis network, including assisting protein synthesis. It will also help to block the pathway that will lead to misfold protein aggregates, and then it will help to do protein degradation if the protein is misfold and aggregate. And within the chaperone family, it has a majority of family belongs to small heat-shock proteins, which is ATP-dependent heat-shock proteins and ATP-independent heat-shock proteins. And I'll bring it to later. And of course, some of the non-heat-shock protein chaperones family are related. And yeah. And now I'm jump to an
Sorry, could you go back for a second?
yeah.
So your figure on the right, you have HSP 100. That's
Yeah.
obviously not
It's not in human genome.
Yeah.
Yeah.
I just wanted to make sure.
I think should I include it because still if a heat-shock protein belongs to like chaperone, so I'm thinking
You just need to mention it in your thesis, right? Just say
yeah.
HSP is not it's not in human. This PDI, Carl and Beck, they are non-heat shock protein chaperone.
Yeah, and it belongs to other chaperone family. And the middle one, Cloud, is a calcium related chaperone, and Beck family basically is another chaperone which is related to apoptosis and autophagy. And PDI is a chaperone that help to form the disulfide bond of the protein for the folding. And yeah.
I don't know. I think stuff like, I mean, I've read, like, I think at the start, PDI and stuff like classed as a chaperone, and bag it is a chaperone. But if you kind of go on the proteostasis network, they kind of classify chaperones separately to folding enzymes. So I don't know whether PDI would be more classed as a folding enzyme rather than a chaperone itself. Equally, bag, yeah, it's kind of a nucleotide exchange factor for heat shock proteins. So it works with chaperones, but it's generally indirectly as a nucleotide exchange factor, so kind of like a co-chaperone. So
Yeah,
I don't know whether you're going to mention at all like chaperone versus co-chaperone and chaperone versus folding enzymes, because you could just classify them all as one if you want, but there is a bit more complexity to that than just one big family.
Yeah. Okay.
Same with HSP 40. That's also technically a co-chaperone more for HSP 70 and also HSP 90. So
yeah. I think these will be some of the small details that I'm going to I have to think about it. How am I going to classify, allocate within the thesis?
So why you classify this HSP 40, 70, and 110 in the protein degradation with proteasome?
I just grouped. I haven't really classified them.
In the left figure.
Yeah.
Here?
No, left.
Left.
Yeah. This is
some of the examples.
Can you point it to the proteasome? Yeah. So that's the involved protein degradation, but HSP 40, 70, and 110, why they are involving in ubiquitin proteasome?
This is what I know from the literature.
Well, how do they prove it?
As review article mentioned it.
You cannot just be careful of when you are citing review. You need to know why.
Okay. Get it.
Yeah, I think with that also, then you'd have things like, chaperone mediated autophagy that you haven't mentioned, but you've just included protein degradation.
Okay.
So this left figure, assistant in protein synthesis. How? Why HSP 90 is assistant protein synthesis?
So basically repair the damaged DNA, and yeah, when the protein or DNA is damaged.
No, maybe sHSP 19 call like a co-localization with the ribosome. It's like the translation mRNA to DNA, and sHSP 19 can convert to the ribosome. Maybe it's assistance like protein synthesis to avoid when they formulate the mRNA, to avoid the mRNA like combined with itself or before they become the protein. Does this make sense? This is sHSP 19 function.
From what I'm aware, though, as well, I think HSP 70 family,
they
have a much larger client list, are generally more involved in like de novo protein folding, so folding newly synthesized proteins. So.
Yeah.
So the left side figure, I'm not sure is actually how did you validate these things? Because while you are talking about DNA damage and HSP 90, that shouldn't have any relevance.
Yeah, I will remake the figures again, and then if to think about it, whether to include in my thesis or not. Okay. And yeah, now I'm jumped to introducing an organ within the human. It's called endoplasmic reticulum. It's basically at least 2/3 of the proteins are going through into endoplasmic reticulum to regulate, to float the 3D structure properly before they have sent to their final destination. And within ER, it has also the function as protein synthesis, protein folding, and protein quality control. And what we know, ER.
If we look at your figure again, How would you translate this on your small figure? Okay, you give a presentation about protein synthesis, protein folding, protein quality, and then you draw a lot of figure down there.
Mm.
Are you going to explain this or not? If you don't, are you going to explain how the protein synthesis, how the protein is folding, how the protein quality is controlled?
You mean in the thesis of the function of ER or?
No, yeah, you talk about function of ER already, right? They are doing protein synthesis, protein
Yeah.
folding, and protein quality control.
Yeah.
And then you draw the figure below and say DNA to RNA, RNA to amino acid chain and protein.
Yeah,
Let's
I
say
think I
in
would.
protein. Are you going to
myself.
explain it in your thesis, or you are not going to?
Depends on the length, and I will read more literature about.
You should have already
more-
idea. Are you going to? That's why I'm asking you.
Yeah.
Are you going to explain
I'll
it?
consider about it. Yeah.
If you are not, delete all these figures.
If not, yeah, I would delete it. Yeah.
If you are going to talk about them, you keep them. If you don't talk about them, you delete it.
Yeah, sure.
Because your protein quality control only write proteasome. The only thing and folding, maybe you should include in folding, unfolding, refolding. Protein quality control in also should include in autophagy as well, not just proteasome degradation. So.
Okay. Yeah, I will. Yeah, need to basically these two slides have to think about it. But yeah, thanks. I see.
I think as well, if you're going to talk about ER, you probably need to distinguish the types of proteins that they actually work with. You said 2/3 of proteins.
Yeah.
So I guess you need to classify what those 2/3 are, because it's not just the whole proteome, it's just randomly 2/3 goes to the ER. It's specific types of proteins, right? So like secretory proteins, say like muscle proteins, I don't believe would get.
Why is 2/3?
Some of the proteins, like they will already go to mitochondria to do protein synthesis instead of ER. So this is one of the examples that I know it. And
mitochondria only have 10.
And I search in literature, not all the protein actually need to be.
Yeah, not all the protein folding in the ER, only the secretory protein folding in the ER.
Secretory.
Yeah. Sorry, my English is not good, but 分泌型蛋白，绝大多数 like membrane protein.
Secreted.
Yeah.
Secreting protein.
Secreting proteins and membrane proteins. Most of them, like 70%, they folding and refolding in the ER. So not all protein folding in the ER, maybe in the cytoplasmic or mitochondria. Yeah.
Yeah, and this 2/3 figure, is that from like a general cell? Because it's probably different between muscle, right? Because muscle proteome will be a lot less ER related, I assume, than a general cell. So I'm not sure the 2/3 would be realistic for if you're wanting to talk about muscle proteostasis.
Yeah, me too.
So how are the muscle protein do this folding and refolding?
Well, they use chaperones, but as Ying just said, chaperone ER is mainly for secretory or membrane-bound proteins. So they're linked close to the membrane, or so they can easily be secreted out. Whereas, if it's just going in the sarcomere, it wouldn't be directly translocated to the ER. It would just be cytosolic or somewhere else.
Wow, that
so
doesn't
the ER.
Yeah, it would be interesting to know.
The ER doesn't fold every protein.
Yeah. Okay, I got it. Yeah, so that would be interesting to know how the actin or myosin they got folded. No. Yeah, it just raised the question for me. It's like, yeah, that's good to know. This is really important. But how? Because in muscle, let's say, like you said, this is a good point, right? Muscle maybe not 70%, not 2/3, but then the things it's like muscle have included a lot of contractile proteins, those things, structural proteins. So how are they folded? Where do they fold it? It would be
I
nice
don't
to
know.
Yeah.
Is there any? I don't know.
No, I don't know
Anyone
either.
knows about the literature?
Exactly. Yeah. So it would be good to know, Jimmy. Have a search.
Okay.
Or everyone, yeah. Everyone who have a chance to read something about that, yeah, have a chance. Of course, muscle will call it sarcoplasmic. It's not endoplasmic. You also need to mention in your thesis clearly. Muscle, they don't call it endoplasmic. They call it sarco.
Okay.
This is very, very important because when you're going to talk a whole things about muscle in your thesis, and then you are always talk about ER. When you go to review or like people read your thing, they will just tell you are doing something wrong because there are no, I don't know if there are all no ER or how the people separate the SR or ER in muscle. There's no clear definition. Based on Sue Bodin's conversation, she also doesn't know. No one knows. She asks everyone. No one actually really answer it.
You mean the difference between sarcoplasmic and endoplasmic reticulum
in muscle?
In muscles.
Yeah. So for clear, another question is, in muscle, do they have ER? We don't know. But we know muscle, they call it SR. So what are the difference between SR and ER? No one knows. Yeah. So if you have if you read through something and people are trying to explain, please let us know.
Okay.
Oh, sorry. I would assume that like muscle proteins are probably folded like in the sarcomere, I would assume. Like as they're getting, it's kind of like they don't need to go to the ER and then be shuttled out. They will just go straight to the sarcomere and get folded by chaperones in its structure. So it is kind of like a localized chaperone mediated folding thing rather than an organelle. I would assume, but probably need to find references for that.
Yeah.
But I think based on like the knowledge that I've read, where it's like, oh, Desmond chaperone or Titan chaperone or whatever, it's chaperones that are cytoplasmic that would probably just go to the protein and fold it in situ in as it's being synthesized and then located to the sarcomere. But that's just my assumption.
Yeah, we'll need a reference though. The other thing is, but you say in muscle is folded in SR?
No.
Cytosolic.
Well, it as you was saying, it depends on the type of protein. So not all proteins get folded in a certain place. It depends on if they're close to the membrane or if they're just in the sarcomere. I'm sure they don't all get folded in the same location. So SR, I mean, yeah, I don't know what the difference between SR and ER are in muscle in terms of if there's an ER specific for folding or whether it's all just globally called SR. I don't think that's necessarily like 100% important because it's still a reticulum protein that folds stuff.
Yeah,
it's just the added thing is the calcium. But.
Yeah. I'm sure even ER they are also doing some calcium things. Just
yeah. I don't actually really know. Yeah, I don't know.
Yeah. Welcome to the ambiguous world, but this is good. This is good for science.
Yeah, but I think my original point is just to make sure that Jimmy's aware that when you talk about 2/3 of protein, it's probably not true for muscle. Or if it is, you need to find out what those 2/3 are because it's not a random 2/3.
Yeah, when it's easy, right, Jimmy? When they are saying two-third, not in muscle, just say two. This is based. You need to write just additional sentence based in other tissue or other cell.
Mm.
Yeah,
Yeah.
may not directly relevant to muscle. Yeah, it's just the way you explain.
Okay. Yeah, I'll be aware of it.
So,
Right.
when you are, writing is kind of disseminate the knowledge. So the only things people try to avoid is to give a wrong message. Yeah, but if you say two-thirds of protein is made in ER, this information may not be directly relevant to muscle, then you are correct. Or you can say based on what cite the literature based on what cell they measure this two-thirds of based on, but this is not necessarily relevant to muscle. You can also say that, okay?
Yeah, in other cell tissue.
Yeah,
Cell
you just need to mention it. Yeah.
Get it. But I'll still have a search on how muscle
Yeah,
works.
sure.
Yeah.
If you can find it, that'll be amazing.
Yeah. Okay. And now, yeah, back to ER. And basically what we know about ER is about proteins. Basically ER's function is basically flow the unflow proteins and send them to final destination. And however, there always have an imbalance of proteostasis or within ER, which is when the amount of proteins need to be folded is more than the folding capacity that ER can be handled. ER stress will be happen, and basically it will occur and a response called unflow protein response and lead to a lot of downstream pathway happening. And the aim of this downstream activation of this pathway is basically try to recover ER into a normal homeostasis condition. And yeah.
You're again, Jimmy, when you are presenting these things, you're just too much shortcut. Go back. When you talk about all this signaling, how are you going to talk about that?
In the literature, in the thesis, I will. And I
you
will.
have to get very detailed, like how IRE1 is regulated.
Yeah.
How they regulate it, how they activate XBP1 and what XBP1 does. How PERK is regulated. How do they activate EIF2A and then ATF4, and then what BIP does, what PDI does.
Yeah. Yeah. Basically, within ER stress or unflow protein response, the first thing that the chaperone does is the BIP will disassociate from these three types of UPR-related proteins, and then IRE1 will firstly dimerize and autophosphorylate, and it will happening into the ER, and then it will basically have two types of in short, basically two types of result. IRE1 decline will be a decrease in mRNA that basically lower the synthesis of mRNA in ER, and then XBP1 is basically a protein that will be sliced in ER and then downstream sent into nucleus and then help to generate some of the proteins that is linked to quality control and which is leading to, or some protein related to associated degradation. And for PERK, similar, it will dimerize and phosphorylated and downstream activate another protein called EIF2 alpha. And it will activate another protein, ATF4 is a activate transcription factor. For this protein, it will basically generate CHOP and lead to apoptosis and also leading to some of the amino acid metabolism and autophagy and ER-related proteins that help floating. And ATF6 is the last main branches of UPR pathway, which in the endoplasmic reticulum, it will basically being cleaved into it will send to Golgi apparatus and being cleaved, and then it will cleave once it cleave, it will downstream into nucleus and also generate some of the increased synthesis more ER-related chaperone or protein, which aim to help to increase the protein folding within the ER. And PDI is basically a chaperone that is around endoplasmic reticulum to do disulfide bond formation of the protein to make them to form into 3D structures. Yeah.
So PDI is actually a good thing. It's not that, so they are helping to refolding.
Yeah, this is what I know at the moment, but the one that I'm testing is a very big family, like HSP90 and HSP90B1. HSP90B1 belongs to heat shock protein 90 family, and PDI is a very big family, and I believe, or what I know in the literature is each PDI family members, they have different functions, which need to read more about it. And yeah, this is what I know at the moment. And this is a simplified version for the UPR protein pathways. And in my thesis, I have made a more detailed pathway graph for each UPR branches, and I've write every single pathways, how it does or how they work, because it'll be quite complicated if I really expand it for each pathways. So this is a simplified version for me to have a note.
Sure, that's fine. So just be careful, you only draw the phosphorylation in IF2A, but not the others. So if you know they are actually phosphorylated, regulated, you should put all of them.
Okay, I will be aware of it.
Because you are talking about, IRE1 is dimerized and autophosphorylated, and there's no phosphorylation.
Yeah, get it. Need to get it. Understand, sir.
And you put a EIF2A. You are biased, right?
Okay.
You know what I mean? Yeah. Why you only give this IEF2 and not the other phosphorylation?
Okay. And yeah, this is in my master thesis. I will basically dip into very more details about small heat shock proteins family. And before I jump into the reason or rationale why I studied, I would like to describe in here what small heat shock protein family does in the proteostasis network. And in general stress condition, basically small heat shock protein will be activated. And firstly, it's in an oligomerized form. And once it having a stress condition signaling, the small heat shock protein oligomers will be activated by disassemble from a very big oligomers into smaller pieces of small heat shock proteins. And then as the stress condition, I'll give an example, will be neurodegenerative disease. You're having a lot of accumulation of misfold proteins. And how a small heat shock protein does is basically they perform a general variholdase function and wait for other small ATP dependent heat shock protein family to come to deal with the unfold or misfold proteins. This is the general function of small heat shock protein that we know. And for
so what's the oligomerization for?
This is the unactivated form of small heat shock proteins. And under stress condition, it will basically disassemble and then bind to the accumulation of misfold, unfold proteins, and then they hold the proteins to prevent aggregation.
So in their functional state, they act alone, and in their inactive state, they bind together.
This is what I know. And then
it's opposite, right?
No, it's this way. I think the oligomerization is more of a almost like storage form for them, and then upon light activation, they sort of like dissociate and either form smaller clusters or individual, just small heat
Is there a
shock.
protein that regulates that, or how does it be or got
So
the
some of them are like phosphorylation activated, like HSPB6. I know that one's got an antibody. I don't know much for the others.
So
HSPB1 as well.
do they all are they all, so let's say there's 10 or whatever, how many there are they all oligomerized in their inactive state? I mean, how do they, is it like certain ones interact with each other?
Yeah, so I think Jimmy's got one on the next slide. I think talks a bit more about.
Yes.
I think
Yeah, the kind of, yeah. So yeah, I'll explain it a bit later, but basically in general it will form into either homo oligomerized or hetero oligomerized, depends on which type of small heat shock proteins they are. And then in general try to hold the misfold proteins and wait for the ATP dependent chaperone heat shock protein family to come and deal with the misfold proteins. And now it will be more specific down to skeletal muscles. As what research or what we know in the literature, that has 11 small heat shock proteins in human genome, and it's only 7 small heat shock protein exists in skeletal muscles. And I've further go down into the literature and found out that in the Sugiyama's group, they actually perform a yeast 2 hybrid experiment, and they found out that basically HSPB1, 5, 6, they will interact to each other, and then HSPB2 and 3, they will add as an other oligomerase group to perform during misfold protein. And yeah.
So this paper, they are not only using yeast 2 hybrid screening, they also do this gel filtration, size exclusion chromatography. Yeah, so I've discussed with Ian about this paper, and this is they are using the more data that support this conclusion is using size exclusion.
Okay, I will read more detail about it, and then this. Yeah, I have a question. Should I mention like more details about how this paper, how Sugiyama makes these two groups?
You can mention it, but you probably don't need to detail because you don't do this experiment.
Yeah, okay, get it.
Right.
What about 7 and 8 that you have there? Do they exist in their own group, or?
Not really.
Because like in your figure here, you've just got 2 groups, but you're still missing 2 small heat shock proteins technically.
Some of the yeah.
How did you exclude other protein? I think you need to write it down.
Like
11 or 10 is only testis, right? And then 1 is the only eye specific, right?
Yeah, 4 is eyes, and I remember 10 and 11 is testes, and then yeah, should I make like an arrow that?
No, you need to write it down.
Oh, I should write it down.
You need to write it in your thesis. How did you exclude other things?
Okay.
Yeah, just like 6 and 8. What Bradley say 7 and 8? Why they are disappeared?
Get it. Cool.
In what reason they disappear? Because
Maybe
like in that review.
Huh?
In that review, I'm just looking at that Tedesco review, just reference. Like why on its figure it says it's only HSPB 1, 2, 3, 5, and 8.
Which one?
I don't know. On the review that you referenced, Tedesco 2022.
You sure? Or
I don't know. I've only just looked at the figure that they've highlighted all the
Okay.
Yeah,
I will.
that's it.
Yeah, I'll recheck about it because this referencing is very, very important. Thanks, Sam.
Just based on the figure they show where they highlight all the. I don't know. I don't believe that because the I detects. I detect more than that.
Okay, I'll research about it. And yeah, I would basically jump to basically our DSM's proteomics data, and in his proteomics data, he used an aged comparison between aged skeletal mouse muscle and young skeletal mouse muscle, and he detected in 7 out of 11 small His proteins in the skeletal muscles, and 4 of them are upregulated. And I found in the literature that basically Sam's proteomics data is same as what literature report in the trend is small His protein basically increase in the aged skeletal muscles, and it's not only one of the there's a lot of group that mentioned that small His protein actually increase in aged skeletal muscles. And same as the ER-rated proteins, it also related. It's also showing the proteomics data that is increase in the aged mouse muscle, and some of the literature also mentioned that ER-rated proteins or chaperone actually increase in aged muscle conditions or related disease. And yeah, this is my hypothesis. And what we know so far is small His protein increase in aged muscle, same as the ER-rated proteins or chaperones. But actually, there has no one really studied the direct mechanism between small His protein and ER stress. And we just hypothesized that under this condition, will small His protein increase under ER stress specific condition as to reduce the amount of mixed protein based on its major function that we know in the literature. And because of that, my master thesis is basically having two chapters. The first chapter will be validation of the antibodies is in ER stress conditions and His condition. And the second chapter will be identify the small His protein response under different time course and dosage amount of the ER stress drug. And
on the previous slide, so previous slide.
Yeah, this one.
So when you say no one studied the direct mechanism between those His proteins and ER stress, is that like in all cells and all like conditioned tissues, whatever, or is this in muscle?
One literature paper mentioned in plant, and other than that, I can't really find it.
You need to mention
I mean,
that.
I would, yeah. You should It's interesting if people have done it in other tissues or other species because there could be some kind of overlap. It's just important to mention that no one's done it in muscle. If that is the case,
Okay.
if you're confident that no one's done it in muscle, that's fine to report. But if people have done it in other tissues or species, then you should report that because it's not true otherwise.
So should I add in muscles like at the end bit of the second point?
Well, you could include any literature that studied the direct relationship in other tissues or other cells if there is.
Yeah.
And just say at the end that if there's no one's done it in muscle, then that's
you need to first
valid.
search if there are any link between small His protein and ER stress or UPR signaling pathway. If there are any link between these, right? In any other studies, and then you make sure if there are actually ensure there are actually no one study the direct mechanism. But when you go direct mechanism, so then you need to know is why you think they have a direct mechanism. In what context? What makes you think they should have direct mechanism? is there any literature, there's an inhibitor of this UPR signaling pathway. Have anyone treat this inhibitor and then measure small HISHA protein expression? If someone does it, means there may be a relationship, but they couldn't show they are direct. And your study is also not going to show any direct, so you cannot say no one study. Your hypothesis there's no one demonstrate there's a linkage or there's an association between small HISHA protein and ER. You cannot say direct because you are not doing direct mechanism. Okay, so your hypothesis has to modify. Yeah.
Okay.
Is that if you say in muscle means there are no one does in muscle, but there are some evidence in other tissues.
Okay.
Does that make sense?
Okay, I'll come back to it.
Yeah, you need to still search around. But this is good, Jimmy. You know, from what you are presenting today, I know you know a lot. Now is the matter. And this is very common. Everyone knows a lot because we read, we do something, we are living, and yeah, we know about how are you going to put those things in paper? That is the things you have to overcome over. Otherwise, I think so far your presentation background is really, really good. But there's one thing I need to mention because your title, your aim of your thesis is going to study tunicamycin and substigagan, and you didn't give any introduction of them.
Yeah, I'm thinking this is the question that I would like to raise as well, because where should I mention the function of them in the thesis?
After you introduce this small HISHA protein, then you can start to see. You must read a lot of literature about these two drugs, right? So you know how these two drugs works, right?
Yeah. So
then you just say, okay, there's a common way they can induce and this and blah blah blah. And why you do this is because have someone does it in muscle or not?
Can you not mention it?
Sorry?
Could you not mention it at this stage in terms of if you've introduced ER and you've introduced heat shock, and then you say, well, there's a gap that in muscle no one studied dissociation, and then could you not then introduce there are pharmacological or I think these are all drugs, right? Ways of activating ER that have been harnessed to study ER stress, and you're basically saying that you've utilized them, studied the response of heat shock. Could you not introduce it at this point, because now you've done the background biology and now you're talking about ways to actually, like the methodological ways to study it?
Yeah, I can. Yeah, when I introducing ER stress, maybe is a good point to bring these two ER stress inducer as well.
It's whether you want to talk about it's well established. There's well established activators of ER and then list them, but then say, and mention how they activate certain parts of the pathway. And you can do that when you talk about all the XPP1, phospholipid EIR1, whatever that stage, because then you can actually directly link the drug to certain pathways.
Yeah. Yeah. Get it. I think I will try to put it into a nice place, because now at the moment in my thesis I just put the function of these two drugs in my methox section, which is not really, I don't think it's a good place. And then
no, it's a good place. You still need to write, but it's just like no problem. It's just you need to, I would say you should have the same amount of detail about these two drugs. In possibly these two drugs, they have already been widely used in other tissues, other cells. Yeah, but what are the common knowledge we will need to know before you move on to muscle? You also show me there are some paper they are using muscle and C2C12 using this tool. So what have they found so far? And how will their information benefit you for doing this research? Yeah.
Okay.
So once you know all those, right? Then you can then once you write your M, then you collect your data, then you can write discussion. Because your discussion is using your result to put into the knowledge you know already. That's basically your introduction. And discuss them, right?
But even I read the ER stress-induced drug state literature used in C2C12, how it helps? Because the aim or the
but if they don't help you, how will they help?
Yeah, this is the question I wonder. Yeah, I will read off.
If you don't write it, that means you ignore literature, right?
Yeah.
Science cannot come out like that, right?
Yeah, but I just wonder. Yeah, I'll read it and then hope it helped when I move to discussion part. But yeah,
the important thing is how you raise. Yeah, how you formulate your question, because your question should be based on literature, right?
Question should be based on literature. Yeah.
Yeah.
Yeah. Okay, I get what you mean. Yeah. Maybe it's a good point. Maybe I'll just mention as well, like literature used the drug on muscles, and no one really study.
Whoa. Literature have using the drugs in muscle, but what do they found? Blah blah blah. But there's no one does blah blah blah. You know this.
Yeah.
So you've identified a research knowledge gap, and then you can do it now.
Okay.
Yeah, but you need to have a good introduction, because whole your thesis is these two drugs and you don't introduce them.
I'm done.
No. Then it's just people cannot understand why. Oh, what? You talk about all this and then what?
Okay.
What is Saptikagen?
Okay. Get it. Yeah.
Jimmy? Sorry, Jimmy.
No, sorry.
You mentioned before, right, in a slide, many papers mentioned heat shock proteins are increased in aging muscle, right? From that one, did you plan to summary in the let's say like one table?
Yeah, I have summary in the one table already. And this is not the way that I presented my thesis. And I have basically screened this paper and list out which small heat shock protein and which animals tissue or human tissue they use in the paper. And yeah, majority is sHSP 1 and 5.
I feel like if you can generate the summary of the table of the previous paper, right, and you can know how many heat shock protein that they found, and then the model that should be include the capsicargan or tunicamycin as well. Yeah.
Okay.
It should add in the introduction chapter. Yeah, for my opinion.
That's a suggestion. This is nice.
Yeah.
A good suggestion. Thank you.
Yeah. I need to search like how would this does. I think I bet people must did it before. I need to search. Yeah. Thanks, JC.
Probably not. You will be the only one.
Maybe. Let's see it. But yeah, I'll search it today and have a look on it. Yeah. And then these are my protein lists and the color, small heat shock proteins, pick it, and then UPR pathways, and I highlight in different color represent the different pathway that represent and respect colors. And for the last bit will be the classical markers that the literature used it for detecting ER stress and chaperone or basically just downstream pathway ER markers. And this is the first chapter. I felt the validation of the antibodies in tunicamycin, pepsigagan, and he shot, and then to show the antibodies are working before I move into the next chapter. And then the second chapter
sHSP7 working. You said to validate the antibodies, right?
Yeah.
So are you validating them as in if they increase in response to the drug or the heat? So sHSP7 doesn't.
You say it's a big cut. sHSP7 what?
Well, this chapter is validating the antibodies. So is it validating them, i.e. they should increase in response to your treatment? Because does sHSP7 increase?
Yeah, it didn't.
But they are different treatment though. How come they are using heat shot here?
To further heat shock proteins. Yeah.
Okay.
Yeah, so it doesn't increase, right?
Increase was heat shot. hSBP1 increase was heat stimulation. hSBP5 as well increasing.
My question is like, what's the point of? Is the plot to show that? Is the validation to show that they should increase in response to the treatment?
Yeah. This is the aim to see it, but
like does.
But Jimmy, your hypothesis for heat shock activation, you expect to see all of the heat shock protein increase, right? Or just some of the heat shock protein increase?
Hypothesis will be all of them because they named it heat shock proteins. And
I don't agree, Jimmy. I think like different heat shock protein have different function. Maybe you can hypothesize all of them increase, but if your results is some of them increase and some of them doesn't change, I think this will be interesting. That means they have specific function in different conditions to the stress, right? If they are all the same function, why cell needs so many heat shock protein, right? Maybe I think all these protein may have their specific function, or they need to like a different heat shock proteins will like some of the complex and 1, 5, 6 or 2, 3, right? Maybe some of them will increase, some of them will not increase, maybe some of them will decrease. This is you do this research, the meaning value. I think maybe not all of them. Yeah. I think this is you can find something different. That's like heat shock ER stress result in some of the small heat shock protein increase, and maybe this small heat shock protein is associated with the ER stress. Maybe not all. Just my thinking. Sorry.
Yeah. Okay.
Yeah, so just Bobby to raise another question. So do you need to have a small heat shock protein there?
Why not?
Because are you doing any heat shock stimulation for your chapter 2?
No.
No, but why say now in his, I think maybe, well, I think I can guess why Jimmy included this data, because in his chapter 1, he need to validate the antibody. How to validate the small heat shock protein? He need to use the heat shock to validate the antibody work, right? So this is why Jimmy include heat shock to validate the small heat shock protein antibody can work.
Okay, so no one against? Everyone happy with this? Okay, good.
Yeah, but I guess then you've got to interpret it.
Yeah. Then you're
So
gonna
I
introduce.
get the rationale for doing this. This makes sense because you want to validate that you're detecting what you plan to detect. It's just how you're gonna explain when things don't go up.
Yeah.
Is it important? I don't know. Like if the antibody is very unspecific, very specific, then you can be confident that it's correct. But I guess you're trying to use the drug to make sure that the band is responding as it should, rather than just doing it in basal level, right? Because if you just did a control, one, you might not be able to see the protein anyway, and two, you might not know if it's detecting the correct band. So I guess your hypothesis was that these bands should increase, and if they do, you can be confident that you're detecting the correct thing.
Yeah. HSP
when it doesn't,
B1 is working well, then we can see. But how about 5, 6, 7? That's what
it might be that like they're not supposed to increase to the same level. It's just important for you to be aware, I think, of like, why doesn't HSP7 increase? Because someone could ask you, well, I would ask you, have you validated the antibody, or are you just losing it?
Yeah, okay. Yeah, so if you are using heat shock to introduce your antibody, you probably need to write another section in your introduction about small heat shock protein can be induced by heat shock.
Well, I don't think you need to do that in the introduction. Surely that's just a sentence in a result. I don't know.
Oh, okay, yeah, that's okay. Yeah, that's right. Sorry. Yeah. And also your why your loading control is GapDH and vinculating?
Should I only include GapDH for all of them?
Yeah.
Okay.
Or vinculating, right? So you just have to choose one.
Yeah.
Jason, what's your question? Sorry.
Yeah, I wonder for heat shock protein from the result, I see they still have the band in the control, right? And then after activate by heat shock, some protein increase, some protein didn't increase. How did you know the antibody is working? Just only see the band and that's it? Or you got my question?
Yeah, I think what suggests each small heat shock protein has different function, and then I need to know how it works basically in shock and muscles, what the trend should be expected to be.
I mean, like I don't know how important it is, but like, HSPB7 already seems quite highly expressed in the control, whereas HSPB1 is not. So HSPB1 seems to be way more responsive to the stress, probably because it's not abundant and it's only activated or it's only expressed under a certain condition, whereas HSPB7 is probably just constitutively expressed to different functions, but is less sensitive to heat. I mean, that based on your results. Like you can still say it's detecting it. It's just it's less efficient for this model because it's already expressed at a high level, so it's probably less responsive to your drug, to your heat. That's what I would comment on.
Okay.
It's not an invalid data. It's just it doesn't match the response you're expecting because it's probably not responsive to heat as much as HSPB1.
Okay.
Yeah.
Because your thesis isn't trying to optimize the response to heat shock, so I wouldn't care too much about this. I just think it's important to note down why you see what you see.
Okay. Yeah, the first chapter is for validation, and the second chapter is using different amount of dosage in two types of drugs and induce ER stress in the cells and under different time course duration. And yeah, I have high dose and low dose, and then in each drug I use basically 3, 6, 12, 16, 24 hours, and then so one drug will be having high dose and low dose, same as another drug, and all the time course are the same. And yeah, this is
Can I give a small suggestion? Sorry.
Yeah.
In this one, I think your font is different size. Be careful of that.
You mean in the graph or?
Yeah, look at your 12 hours and 24 hours. The font size are different.
Yeah, I'll adjust it. Thanks, I see.
Yeah.
When I put it on the thesis.
Yeah. You don't want your reviewer to pick up this kind of small things.
Yeah, this I don't really like it. So yeah, thanks for reminding me.
And they will just see like, yeah, wow, I can see this student already.
Yeah. And then, okay, these are the representative blocks that I have at the moment, and I have still some of the batches need to be imaged and not redo to do it, and I need to wait for a while. And this is the Fabsigagan representative block that I have at the moment. As we can see, small HR proteins 1 and 6, they would basically decrease general under ER stress conditions. Meanwhile, 5 and 7 don't really. And on the right will be UPR stress response figure. And yeah.
Yeah. So here is very important, right? So when you talk about it, so like before, I would see that HSPP1, HSPP6 are actually decreasing under ER stress. So the hypothesis, your hypothesis was thinking they should increase, right?
Yeah.
So like what GP had just mentioned, what is your hypothesis? You need to write it. So your hypothesis were going up. But when you actually tested these, they're going down. All right, good. So you're gonna discuss why it's going down and not going up.
In the discussion part, and then with the literature support and show my thinking pathway.
Yeah.
I don't know whether it's just come to my mind, and I don't know whether this is at all relevant, but it's probably relevant, but whether it's actually happening is, so part of the UPR pathway is activate the unfolded protein response. It's activate the unfolded protein response and activate certain proteins that will help towards protein folding, right? So like BIT, HSP90, whatever. But generally, another angle is that it reduces proteins translation.
Samuel, you might to speak again a bit slower. Yeah.
Okay.
Thanks.
So with your condition, you're activating ER stress. ER stress activates the unfolded protein response, and that has different angles that it functions with. One of them is to increase ER chaperones like HSP90B1 or BIT or PDI, right? But part of it is to reduce global translation, like to reduce synthesis, to try and stop the stress from becoming too much. So could you argue that the reduction in proteins that you see is just a result of general global inhibition of translation in the HSPB stuff?
Can you repeat the last bit again?
I also didn't get everything. Anyone who get everything?
I got this. Maybe some hypothesis is that like the ER stress will result in like the phosphorylation of the F2 alpha will result in the reduce of the general protein translation, right? So some said that maybe this will result in small HR protein decrease, right?
Yeah, the idea of my hypothesis would be if most proteins are being reduced, i.e. the translation of most proteins is inhibited, could the heat shock just be a by factor of that? So they're not like directly reduced because of actual folding capacity. It's just because they don't go under the category of, oh, we need to increase these proteins, so actually we'll reduce them instead.
But let's go back to the mechanism graph, Jimmy, go back to. But I think one of the ER stress, like the translation factors, will result in the champions increase protein. sHSP.
Yeah, but I just don't know which, like, if no one studied it, then no one knows which chaperones are increased, right? If no one studied it, this is by Jimmy's introduction, this is the first study to do so, right? This is the first study to look at heat shock. So no, if I'm trusting Jimmy, no one's done this. So you can't assume that heat shock are part of that. Sorry, you can't assume the small heat shock part of this increase. Now you're right. Hypothesis, I would agree that you would expect maybe all chaperones to increase, but based on the results, they don't. So could the other hypothesis then be, well, maybe they're not part of this specific cluster of chaperones that go up and they therefore go down because they haven't been selected essentially as, oh, we need more of you, so we'll just have less of you, right?
I agree.
Could that be the result?
Yeah, maybe. I agree.
I think it's worth thinking about because it came to my head as like, well, you're only blotting heat shock proteins. So you're only blotting small heat shock proteins or the unfolded protein response. You haven't blotted for like turomycin or like anything that looks at global reduction. And maybe that would be interesting. I'm not necessarily saying you have to do this, but it's something to think about in that if most protein synthesis is reduced, as you would expect based on the pathway, could you just argue that it's just a byproduct?
Yeah,
could be one explanation. Yeah, very good explanation. Yeah.
I just think it needs to be mentioned now. I think that's probably.
But how come
definitely a hypothesis that has legs.
There are some protein that also increasing like BIP, IRE1.
Yeah, because they're directly like they've been proven to act like studies have measured this. So they have been proven to be part of. But what is known is that this increased translation is selective, right? Because the whole point of ER stress is we need to reduce global translation because there's too much stress on folding. So most proteins are reduced, are increased. So maybe the heat shock, small heat shock, aren't part of that selective increase. So therefore, you would expect it to go down. So it's not maybe surprising in that sense.
Yeah.
Because maybe the stress isn't so much at this stage that all this aggregates. So therefore, we don't need you right now. So we'll reduce you.
Yeah.
That would be my.
That's good. Yeah, that's a very good point.
I wonder how can I measure the protein synthesis with pyromycin, right? Is it an antibody or?
No, you'd have to do an experiment, like an incorporation experiment. And it's just something to note. I don't know if you'll have time to do it. It's not long, I don't think, but yeah, it would be interesting, maybe.
You can measure protein. I don't know if you read the paper did you read? No. At least Bradley and In should have read the paper I sent over about Journal of Physiology.
Label the protein synthesis, right? Label the protein and can.
They are using mass spec, so they can specifically looking at specific protein.
Yeah.
That technology you can do. It's a Benjamin Miller paper. Yeah. But they also argue that in the way that right now we measure protein synthesis in the lab using radioactive or JC had been measured. So Jibisu had been and Bradley, they were been learning from the medical school. There's a caveat because they are just measuring some of the high turnover protein. But we are measuring all protein synthesis to say, okay, all the protein synthesis is going up, but that's not right. That review clearly argue that one, but that's absolutely correct. Don't know if you understand, but yeah, maybe I can share the paper again to everyone because that's a good paper to read a bit when you're going to discuss this.
Yeah.
Yeah.
Sure. Let's send it over.
Yeah, after some hypothesis, I rememorize that all the Hayshak proteins translation factor is HSF1. So Jimmy, you can find the literature whether there are some association and link between ER stress and HSF1. HSF1 is all the small Hayshak protein translation factor. Yeah, I think if you can find some link, maybe ER stress have impact on the HSF1 and then the Hayshak protein, small Hayshak proteins, protein or mRNA level change. Yeah, just like some hypothesis. Yeah.
Yeah, it's worth to read it. Okay.
Okay, cool. So this also raised another question, right? Because when you are doing this HeSHA protein, you see your small HeSHA protein is increasing. Have you tested using those HeSHA protein looking at UPR's pathway?
Who?
You know, look at your previous chapter 1. So your chapter 1 shows HSPP1 is increasing, like aging. HSPP5 is increasing, I would say. Yeah. I doubt because your quality is not 100% convincing me. Okay. HSPP6, not sure. HSPP7, definitely not changing. Okay. Not increasing anyway. So have you blocked this sample for any of the UPR, like your left side?
I haven't. You mean for his shot, right?
Yeah. Because this will tell you now here, your data will be very, very clear, tell you if any UPR signals are relevant to this small HR protein expression. If they are, they should be the same. If they are not, they will be different.
Let me. I really forget whether did I block the ER markers or not. But I think I have it, but not all of them.
So for example, if you see none of the UPR protein is increasing under the HeSHA stress, and then you see HSPP1 increasing with HeSHA, and then here you see all the UPR protein is increasing with the substigagan tunicamycin, but your HSPP1 or HSPP5 protein is actually decreasing. That is very, very clear evidence to telling you UPR signaling doesn't regulate HSPP1.
Yeah. Worth to. I get what you mean. Yeah.
You know, doing science is very easy to disprove them. It's not easy to say they regulate this, but it's easy to say they are not.
Yeah, I'll search it. Yeah, I think I have blocked some of them. Yeah, and this is the representative figures for the tunicamycin in chapter 2, and it's also show that 1 and 6 small HSPP 1 and 6 decrease over 2 types of dosage treatment in tunicamycin, and yeah, UPR response is a bit different in tunicamycin compared to fepsigagan, but this is the figures that I have at the moment. Still, some of them need to be, the batches need to be redo, but I need to be wait for a while. And
you explain what's are the different?
I would say IRE1 doesn't increase in tunicamycin treatment, and ATF6, the click form is different, and EIF2 alpha is different when tunicamycin compared to fepsigagan, and Yeah, the activation time of different UPR pathways is different between two drugs. But
BIF2 is very tricky because you increase in third hour and then they decrease and then they increase again.
Yeah. So
is it consistent for 3 of the experiment?
Yeah, and then I just need to be redo and
they are consistent. They go up, down, and up.
Yeah, it's not happening in Fepsigagan, I would say.
I know. I mean, tunicamycin, you have done tunicamycin 3 times.
Not 3 times yet.
3 times it go up and down and up?
Not yet. I need to wait for the box.
Okay.
Yeah. So this is the data I have at the moment, and yeah. I'll prove whether the pathways are really having this trend or not once the deer boxes got fixed. And then yeah.
Yeah, cool.
Yeah, it's
okay. Move
at
on.
least 2, I would say at least 2 batch. I remember at least 2 batch are having this trend. And then I'll see the last batch is having this trend or not. So yeah. And now is the little question that I'm like around my head for these 2 weeks. Basically, I have 12 proteins in total, and now I have 4 conditions, which is high dosage and low dosage in one drug, and I have 2 drugs. So in general, if I really following the figure on the right, one dosage, one graph to represent one protein in one dosage, I'll have basically 48 graphs, 12, 12, 12, 12, which I don't think is possible to do it on my thesis. And
why not?
Or why I need to do 40? I mean, that's not a good way to present it, right? And then
it's a good way.
You sure?
Yeah. But you can separate small HISHA protein and
And I want to know, like, does anyone have a good advice? Because this is the first time for me to really handle the statistics, and as what Wasi mentioned, I've also think about, should I bring all the UPR specific markers, like or per pathway, I have 3 proteins. Should I bring them all into the same figure and small HISHA protein bring into the same figure as well as for presenting one image? Because I just think 48 graphs would be too much, and then
it's not. I think it's normal. Yeah.
I have no experience before, so I wonder how you guys think about it.
I think if you do small HISHA protein, you have only 4. Both small HISHA protein 4 is 16. And then you can have 1 representative plot and then have 1 figure.
For 1 representative block for one for each small HISHA proteins, and 4 statistics figure.
Have 1 representative block for 1 drug.
Yeah.
Right. sHSP P1, B1, B5, B6, B7. And then you have a statistic for 1 drug. 1 dose, 1 drug.
You mean, okay, tunica mouse and high dose, and then bring all the small HISHA protein together.
Yeah.
Okay.
And the same for UPR.
And the UPR will be presenting specific separately, right?
Yeah. I would do that. Yeah.
Okay. Yeah, I'll get it. So, but
if you look at this like a Nature paper or thing, they have just like a small figure A, B, C, D, E, F, G. What's the problem with F, G, H, I, J, K?
Get it. Yeah, just thinking about how to present it in a better way. But yeah.
Is there any other suggestion? I think for your left side, if you do bar chart, it's better, and then you can still put a three sample value individually into each bar. No? Yes, you can do that, yeah?
3 sample into each bar.
Yeah. You can still have a dot in each bar. No?
Yeah. They was make a very first trial.
And including a standard error or standard deviation, standard error. Yeah.
Okay, and then I would do it separate the dosage, right? Like only
Yeah.
do low dose in the figure, and then
Yeah, low dose, and then you do high dose immediately, and then low dose, high dose.
So separate into 2 graphs for UPR, and then separate out into dosage, and small heat shock protein will be grouped to present together according to dosage group.
That's what I would think. Is there any other one have any better suggestion?
Okay. Yeah. I'll make it up. Yeah. Yeah, I think this is the question that I have at the moment, and as long as I got all the figures, and I'll move on to making a statistic graph, and yeah, this is what I'm going to present it today, and yeah, thanks guys.
Do you have any main conclusion?
What conclusion?
What did you find in your work?
I haven't made it yet.
Yeah, you need to start to list it. Like what Sam does right now. Just he lists and then start to write each section about your discussion. Formulate your section of discussion.
Okay.
So you remember last time when we discussed some stuff, and then we talk about what is your finding each from this study. Then you can list 1, 2, 3, 4, 5. I don't know how many you find, and all of them. And then the most important one go first, second important one go second, and third. And then you can. It's just helping you to write your discussion more faster, and more structured.
I'll try to make a list out. Yeah, good idea, YC. Thanks.
No.
Yeah. Basically.
Amazing. Yeah. Jimmy, I think you learned very much and very well.
Thanks. Yeah.
Cool. I think what is very dangerous things is you should try to focus on the work. When you are writing, you need to focus, write down something that are relevant to your title and your aim and your result. Don't write too much about what you already know.
In the discussion section. In the introduction.
Yeah. Because like, for example, your figure 2, your slide 2 and 3, you can put them all together in one paragraph or 2. Each paragraph should be like 5 sentences, not more than that. Figure 1 particularly. Slide 2, keep it very short.
1 page maybe, and then go.
Oh, probably half page will be enough too.
Yeah, get it.
Any other suggestion?
I think for introduction, Jimmy, you have to have like a territory to tell what is the thing that you want to include it. Is what is the gap that you found. You know what I mean? Like, because when you present, I found a lot of data, but I didn't see the specific detail that mentioned why you study this heat shock, like a strongly support that why you study this heat shock.
Strong support.
Yeah.
You don't have a strong rationale why you study this small heat shock.
Yeah.
Yeah, I'd agree in terms of you introduce proteostasis, but there's like so many branches to that. So you probably need to make it clear as to why you focus on this question in particular.
Okay.
So the introduction need to guide people to be able to understand why you measure this, right? Basically.
I mean, are you trying to, I don't know, is your rationale purely based on my data?
No, I think he has included some other things as well, so it's good. Yeah, mainly is because of your data, of course, but when you are writing, different.
Is the rationale about aging or not? Like, is this an aging introduction or not? That's what I get a bit confused about in this. Like, is it really related to aging or not? This stuff.
Yeah, that's depending on how you're gonna write. Yeah. You can write it as an aging standing point, no problem. Just say we saw this in this, and then literature also support all this finding. So in order for us to do our first hypothesis, since aging is related to ER stress and things, so then we are using chemical inducer to see whether this can mimic aging. Just do it. And of course, the hypothesis is going to be increasing similar, but it's not, right? Why is not, then you discuss. Does that make sense?
An extension study from the aging data, but not really studying about aging. This is what I think.
But I think for Jimmy's data, I don't think the rationale can extend to aging. Yeah, I don't.
You cannot, but you can say, because of we have this hypothesis, we find out that in a skeletal muscle with things, and then therefore, and literature also support, which demos this. So the data from, let's say, step by step. So some data shows us that there's a ER stress, right?
Yeah.
Yeah, and then
I guess
we can use the
right.
ER chemicals to induce ER stress to see whether this is the way can be mimicked, but your conclusion is not, right?
Yes, the other angle though is like, yes, aging data supports that the ER stress goes up in males and heat shock proteins, small lesion proteins go up as well. But would it be a bit naive to just focus on aging and not mention any other atrophy models that show the same thing? Because at the end of the day, this isn't an aging paper. I don't know what your thoughts are in terms of like, like, are there any models of muscle deterioration that also show the same thing? And should we really be just in the introduction, should Jimmy just be focusing on aging papers? Or should he also include like, denervation or other muscle disease related criteria that also might show the same thing? Because at the end of the day, this isn't an aging paper. So
yeah, you can include
would
it.
It be smart to include, like, not just narrow it to aging in the introduction, but try and broaden it to muscle related?
Yeah, that's a good idea. Yeah, you can have a paragraph related to muscle atrophy model that relevant to UPR or ER stress. Yeah.
Okay. I'll try to search it. Yeah.
I just don't. Yeah, I don't think necessarily you could go about it the way you've done, where your introduction is like at the start it's aging. What does aging show? But because it's like a mechanism, it's probably broader than just aging. So you could probably find papers in other muscle-related diseases that show ER stress going up or small lesion proteins going up. And if there is, maybe you shouldn't limit yourself to just aging.
Yeah.
I don't know.
Yeah. Alternatively, you can also say, like, because this drug, they are activate ER stress with different function, right? So you can test whether any of these drug like regulating glycosylation protein, it is one of the mechanisms that cause aging. You can say no. Your result shows no, right? Or ER stress is relevant to aging things.
So you can't use this data to support any aging.
Yeah,
hypothesis.
I don't.
No. But there's this data I can tell.
I think from Jimmy's data is simple. Like, from some data, aging will result in ER stress and a small H-sharp increase, right? And ER stress and the small H-sharp protein are associated with the proteostasis. From Jimmy's experiment, just to test whether we induce ER, it will change the small H-sharp protein changes. They are all associated with aging. Just from Jimmy's data, just like he induced ER stress, whether this small H-sharp protein changes with ER stress, UPR pathways. If they don't change, maybe they are from different factor. Aging will this 1-2 factor increase. But if Jimmy's data doesn't say this, that means ER may be not the reason results in small H-sharp protein changes in aging.
Yeah.
This link.
Yeah.
But I still don't know whether is going back to JT's comment on the importance of what you're doing. The broad importance is, are these processes involved in disease and health, right? That's kind of like the first paragraph of like, why are we studying what we're studying? And at the moment, it's narrowed to aging. And you could do that. But if we were trying to put the importance to just like health, then, or muscle deterioration, then maybe it's more important to just like list a few different, factors, whether it is mice models or human models or whatever, bed rest or whatever, where people might have measured ER stress going up, or even at a Western blot, and that's more of like we know these go up under stressful conditions in muscle. Both ER and small heat shock. You could focus on aging if you want, but I just think there's probably more to it. Because your study then isn't really focused on aging, so you don't need to limit yourself to aging.
Yeah, that's a good idea.
Like what? Yeah, I would ask, why are you ignoring?
Yeah, because your
going up under injury. Maybe it goes up under injury.
Yeah, your introduction also talk about diabetes or something. I don't know. Yeah, you can also include those things. Yeah.
No, reformulate a bit, but I get what Sam tried to suggest. Yeah. Thanks, Sam. But yeah,
all you're trying to do is tie two mechanisms together, but the mechanisms might be involved in loads of different muscle-related conditions. So why not introduce them all?
But if I introduce, will be in the very beginning, right? Like here.
Your beginning is already. Yeah, you also talk about diabetes, cancer, right? So.
Yeah, but this is just the reduction of muscle, not the impact the ER stress might have. Because then further down the line, you mentioned how ER stress goes up after aging, and you list all those papers. But
ER stress.
I guess my question: you could focus on aging. It's fine to do that. I just wonder whether you want to take other angles as well.
Yeah, I'll have a search on it, and then I think it's good to like mention it in the introduction as well. Okay. Get it.
Cool. Yeah. So from this talk, I think I learned a very important thing is the small H-sharp protein complex function, because like it might be, and this is also a similar question, when Sam is reporting his data in this conference. This Japanese guy, he was asking him, can he look at their complex or stress? I think, apparently these complex things are really interesting things to do. And from the paper, they are talking about is to look at their complex. And what Jimmy has told us today is usually the small H-sharp protein is forming a complex when they are not functioning. And then they dissociate from the complex to hold a protein when they need to work. So, this is what I have done today. So this So this is something that people has not done under stress.
Under ER stress, yeah.
Under ER stress, yeah. Under aging model. In the aging model, no one has done that. And because that paper,
No one has done an aging model about.
the paper you share with me and we will discuss this the other day. And this Japanese group, they have been using this two hybrid system gel filtration to show these things. I think this will be very interesting experiment to do under different conditions.
Let's say what
Not
kind?
yeah. That's okay, Jimmy. It won't be in your thesis. I mean, this is just the things I learned from here. So it means your talking is very valuable. Thank you very much. Yeah. So people who think about this, we can think about this because we can do this quite easily. But we need to focus in a model to induce ER stress and look at their complex. All right.